Hello students, how are you? Kya hal chal? Students, today we are going to discuss a very important and interesting topic from NCERT 11th class biology, that is cell, the unit of life. This chapter has several basic concepts. If you learn these concepts, it will be very easy to understand the rest of the chapters from NCERT biology. So think that you are in an offline class. I am your real teacher standing before you and you are sitting before me in the bench, and take down each and every point which we are going to discuss today, students. At the end of the session, I will assure that you will be able to solve any type of question which will be asked in the NEET examination from this chapter, cell, the unit of life. So let us start the chapter. What is a cell? Cell kya hota hai? So a cell is a fundamental, structural and functional unit of all living organisms. A cell is fundamental, structural and functional unit of all living organisms. So whatever living organisms we see around us, maybe the plants, maybe the fishes, maybe the birds, or maybe the human beings. So all the living organisms which are found around us are made up of the fundamental unit which is called the cell. So just like a large building which is made up of small units called as bricks, all the organisms are made up of the fundamental units called as cells. Understood the analogy? Okay, so copy down this point, students. Cell is fundamental, structural and functional unit of all living organisms. Okay, so next, all living organisms are made up of cells. Next point, in multicellular organisms, cells show division of labor. What are multicellular organisms, students? The organisms whose body is made up of several cells are called multicellular organisms. Just like your hydra. In the hydra body is made up of several hundreds of cells. The cells are present all over the body, in the tentacles, in the mouth, in the body, in the base, everywhere cells are present. Okay, so these cells, they have division of labor. They show division of labor. What is division of labor? Some group of cells, they take the task of digestion. Some group of cells, they will take the task of excretion, and some of the cells will carry the function of respiration. So like that, whatever the living activities are there in the living organisms, they will be shared by a certain group of cells. This is called as division of labor, a very important character which we can see in the multicellular organisms like your hydra. So what is a hydra? It is a multicellular organism. Multicellular organism. And our human body is also a multicellular. We are also multicellular organisms. In our body, group of cells, some group of cells will carry out certain function and some other group of cells will carry out certain function. For example, nervous cells, the nerve cells will carry out the function of transmission of nerve impulses, whereas the cells which are present in the kidneys will carry out the function of excretion. Understood, students? Understood? Yes. Next, in unicellular organisms, a single cell will carry out all the functions. Unicellular organism, example, amoeba. Amoeba. The unicellular organism example is amoeba. Another example is your paramecium, euglena, etc. So in these organisms, the body is made up of only one cell. That's why they are called the unicellular organisms. They are obviously microscopic, very small. Since their body is made up of only one cell, the single cell should take care of all the functions such as the digestion, excretion, respiration, reproduction, etc., etc. So there is no division of labor in unicellular, but there is division of labor in multicellular organisms. Understood? Next, let us go to the next point. For first time, students, Robert Hooke, a scientist called Robert Hooke, he discovered the first plant cell under the microscope The microscope was invented by himself. This is the microscope of Robert Hooke. Under this microscope, Robert Hooke साहब ने पहला plant cell को देखा. Okay, so he actually observed the cells of cork tissue of a tree under the microscope. In that cork tissue, he have observed several small chambers or compartments. So these compartments, he called them as cells. They look like honeycomb. Understood? So that was the first cell which was discovered by a scientist called Robert Hooke. Later, Anthony von Leeuwenhoek, another scientist, he discovered the first living cell under the microscope. Leeuwenhoek also constructed his own microscope, which was very powerful in magnifying the objects. Under that microscope, you can see here, this is Leeuwenhoek's microscope. Under this microscope, Leeuwenhoek observed a water drop from pond. He observed pond water drop. So in that pond water drop, he have observed the first living cell, such as the Paramecium amoebas. Okay, so like this, the first living cell has been discovered by Anthony von Leeuwenhoek. Leeuwenhoek साहब ने पहला living cell को discover किया था. उसका बाद Robert Brown, another scientist, he discovered a cell organelle inside the cell which is called as nucleus. So whatever the structure, cell organelle called as nucleus, which exists inside the cell, this nucleus inside the cell was first discovered by a scientist called Robert Brown. Students, I have something very interesting to show you. So I have an onion and a simple pocket microscope. I will go and get it. An onion and a pocket microscope. So I can show you the cells which are present in this onion by using this pocket microscope. So in order to see the cells, first of all, you should peel the outer layer of this onion. So once you peel the outer layer, inside you can see the onion membrane, and I am going to put this onion membrane under the microscope and I will show you the cells. This is the onion membrane, you can see. This is the onion membrane. Can you see here? Now I am going to keep this delicate onion membrane carefully under the microscope. I am sticking to the microscope. You can see here, I just kept the onion membrane under the microscope. Yes. Now I have mounted the onion membrane under the microscope, and I have some adjustments, couple of adjustments here, and I'm going to adjust it. Wow, I can see beautiful rectangular shaped cells of onion. Wow, wow, wow. You want to see? Wow, look here. Now let me make some adjustments, little bit. Yeah, yes, little bit. Yeah, now you can see the onion cells under the microscope. You cannot see any nucleus or any cell organelles because I am just using a very simple basic type of microscope. I am zooming a little bit so there will be a closer view of the cell. So these are all the onion cells, students. Amazing, right? So that was an amazing view of onion peel cells under this small pocket microscope. Let's see who created this funny microscope. Made in China. China people has created this. So funny, right? Very handy for observing small biological samples like pollen grains, insects like this. Now let us enter into the topic, students. Onion peel cells. Onion peel cells are plant cells. Okay. So here you can see several rectangular cells. They are arranged side by side without any gaps, intercellular spaces. So each cell, each rectangular shaped cell is an onion cell. So the cell is covered by a boundary which is called cell wall, which is called as cell wall. So every plant cell is covered by a boundary that is nothing but a cell wall. It is made up of material called cellulose. Okay. Below the cell wall, actually there is a membrane called plasma membrane, but in microscope we didn't see that. Why? Because the plasma membrane being very thin, it is tightly pressed against the cell wall. So it is very difficult to see the plasma membrane under normal microscope. Why? Because the plasma membrane gets tightly pressed against the cell wall. But actually plasma membrane is also present in the onion peel cell below the cell wall. And next, what is present under the plasma membrane? There is lots of liquid which is called as cytoplasm. Cytoplasm. Okay, it is a liquid that is present inside the plant cell. So this cytoplasm is the main place where several cellular activities will be happening inside the cell. So in the cytoplasm there is a very important organelle which is called nucleus. Nucleus क्या होता है बेटा? It is the brain of the cell. It will control all the activities of this cell. So each and every onion cell is having a nucleus in it. Okay, when there is a single nucleus inside a cell, it is called as uninucleate cell. Okay, so all these are uninucleate cells. Every cell is having a single nucleus. Okay, so this is about onion peel cells, which we can see very easily by using a small microscope. Then what if we want to see an animal cell? So we can use our own body to see the animal cell because we are animals. So here we are going to see cheek cells. Cheek cells as an example for animal cells. Then how can we collect these cheek cells? Simple, students. Take a wooden spatula, okay, a ice cream spoon, something like that. Thoroughly clean it, and afterwards insert that ice cream spoon into your mouth and scrape the inner walls of your cheek. When you scrape the inner walls of your cheek, then what happens? The epithelial cells will stick to the spoon. Okay, then apply that extract, apply whatever you have collected from the inner side of our mouth on a clean slide and stain it properly and observe under the microscope. When you observe under the microscope, you can see these beautiful cells of cheek. Okay, these are cheek cells. Okay, so you can also see the picture of cheek cells here. This is the picture of cheek cells, and this is the picture of onion cells which are photographed under the microscope. Okay, so when we observe the cheek cells, what details we can find under the microscope? First of all, there is the outer delimiting membrane which is surrounding the entire cell. It is called plasma membrane. But where is cell wall? Cell wall is absent in the animals. We don't find cell wall. So there is the outer delimiting membrane called as plasma membrane. Below the plasma membrane, you can find lots of liquid which is called cytoplasm. It is the main arena of cellular activities. Several cellular activities will be happening inside the cytoplasm of animal cell as well as plant cell. Okay, and obviously there will be a nucleus. This is the nucleus, the brain of the cell that controls all the functions of the cell. Now inside the nucleus, what is present? What is present inside the nucleus, children? So inside the nucleus there is a very important material, that is DNA, deoxyribonucleic acid. It is the genetic material. It stores the genetic information. DNA is present. So you are aware of the shape of DNA. Okay, the DNA will be like this. This is the DNA, a very long molecule of DNA. Several long molecules of DNAs will be present in the nucleus. Okay, when the cell is in resting state, we find the DNA finely dispersed throughout the nucleus. But the moment when cell starts to divide, at that time the DNA, it gets packed into rod-like structures which are called as chromosomes. So in human beings, how many chromosomes we can find? Come on, tell me, students. 23 pairs of chromosomes. How many chromosomes are there in human being? So in any human being, every cell in the nucleus, we can find 23 pairs of chromosomes. 23 pairs of chromosomes. Very important general knowledge question from biology students. In human being, every cell will have 23 pairs of chromosomes, that is equal to 46 chromosomes. Okay, so this is about the cheek cell in human beings. Okay, now let us discuss the concept of cell theory, which was proposed by two scientists, Schleiden and Schwann. Schleiden and Schwann were two scientists. Schleiden, he belonged to Germany. He is a German botanist. Schleiden, he studied several plant tissues, and he told that all plants are composed of different types of cells which form tissues. And Schwann, he belonged to British. He was a British zoologist. He studied several animal tissues, and he told that the animal cells have a thin layer called as plasma membrane. He studied animal cells. He told that the animal cells have a thin layer called as plasma membrane. And also he studied plants, and he told that cell wall is unique character in the plant cells. Presence of cell wall is unique character of plant cells. And both of these two scientists, they proposed the cell theory. So the cell theory states that all living organisms are composed of cells and products of cells. So all the living organisms are composed of what, students? Cells and products of cells. This was the theory given by Schleiden and Schwann, which is called cell theory. So you can see the pictures of the two scientists, Schleiden and Schwann. Schleiden, the German botanist, Schwann, the British zoologist. So these were the two scientists who proposed cell theory. So however, the cell theory proposed by Schleiden and Schwann has a demerit. What is that demerit? The cell theory couldn't explain how new cells are formed. नया सेल्स कैसा पैदा होता है, इसके बारे में कुछ भी जानकारी नहीं है इस सेल थ्योरी में. इसीलिए दूसरा साइंटिस्ट Rudolf Virchow, another scientist, he filled this gap which is there in the cell theory, and he added one more line, that is omnis cellula e cellulae. Omnis cellula e cellulae means all cells arise from pre-existing cells. He told that all the cells which are existing, they are formed from the pre-existing cells. So this completes the whole cell theory. So now we can add the first statement given by Schleiden and Schwann and the second statement given by Rudolf Virchow. So together, if you add these two statements, it will fulfill the cell theory. Okay, you can see the picture of Rudolf Virchow here. He is Rudolf Virchow. So this is about the cell theory, students. Now let us discuss another topic. Students, now let us discuss the concept of prokaryotic cell and eukaryotic cell. So the living cells can be divided into two types based upon the type of nucleus, that is prokaryotic cells and eukaryotic cells. So what is karyon? First of all, what is karyon? The term karyon means nucleus. Nucleus. This is also name of our YouTube channel, Nucleus Neet coaching, right? So those cells which have a primitive nucleus are called prokaryotic organisms, and the cell is called prokaryotic cell. So those cells which have a well developed nucleus, a true nucleus, it is called eukaryotic cell, and the organisms are called as eukaryotic organisms. Okay, so based upon the type of nucleus, the cells can be divided into two types, prokaryotic cell and eukaryotic cell. Now let us discuss prokaryotic cell. Prokaryotic cell. Pro means primitive, karyon means nucleus, a primitive nucleus. Okay, so prokaryotic cells are found in bacteria. The prokaryotic cells are found in bacteria which belong to kingdom Monera. Okay, so a typical prokaryotic cell, if you observe under the microscope, so a typical prokaryotic cell, it appears like this. A prokaryotic cell is covered by a rigid cell wall. It is covered by a rigid cell wall. So below the cell wall there is a thin membrane called the plasma membrane. Plasma membrane. a thin membrane is present. Okay, so क्या है बेटा? The outer rigid layer. The outer rigid layer is cell wall. Cell wall. Just like plant cell, the prokaryotic cell also have rigid cell wall. इसके नीचे क्या है बेटा? इसके नीचे है thin membrane which is called plasma membrane. Plasma membrane. Okay, so इसके नीचे क्या है? बहुत सारा liquid. Cytoplasm. What was that? Cytoplasm. Cytoplasm. It is cytoplasm. Okay, so now in the cytoplasm there is no nucleus. Nucleus is absent, but the DNA, deoxyribonucleic acid, the DNA molecule, it is compactly folded and arranged in a region. The region where you can find the DNA, this region we call it as nucleoid. It is not nucleus, it is called nucleoid beta. Nucleoid क्या होता है? So this is the region where you can find compactly folded DNA. Circular DNA is compactly folded and packed. This region is called as nucleoid. It is not covered by any kind of membrane. So that's why they are called prokaryotic cells, because nucleus is absent. So this is one of the important character of a prokaryotic cell. Then eukaryotic cell, U means good or true. A true nucleus or well developed nucleus is present in these organisms. Eukaryotic cell, if you observe, an eukaryotic cell, it has cytoplasm, lots of cytoplasm liquid. In that there is a well-defined nucleus. What is there? A well-defined nucleus पाया जाता है eukaryotic cell के अंदर. Okay, so the nucleus is the brain of the cell. It acts like a brain of the cell. It controls all the activities in the eukaryotic cell. Inside the nucleus, what you can see, there is a liquid called nucleoplasm. We will be learning detail in the next topic. So right now, briefly I am telling you, inside the nucleus there is a liquid called nucleoplasm. In that nucleoplasm, there is compactly arranged DNA fibers which are called chromatin fibers. These DNA fibers are called as chromatin. Chromatin fibers पाया जाता है nucleus के अंदर. So what is this? This is a compactly folded form of DNA, deoxyribonucleic acid, and the nucleus is covered by a membrane. What is that membrane? The membrane is called nuclear membrane. चलो बच्चों मेरे साथ बोलो, nuclear membrane. Nuclear membrane. The nucleus is covered by a membrane called nuclear membrane. क्या इस prokaryotic cell में nuclear membrane है? नहीं है. Nuclear membrane is present only in the eukaryotic cell. Okay. Now let us see some more differences between the pro and eukaryotic cell. In prokaryotic cell, there are no cell organelles. Cell organelles absent. Cell organelles absent. जैसे माइटोकॉन्ड्रिया, एंडोप्लाज्मिक रेटिकुलम, गोल्गी कॉम्प्लेक्स, लाइसोसोम्स, इस तरह cell organelles are absent. But whereas in eukaryotic cell, different types of cell organelles are present. मौजूद है, बहुत सारा cell organelles मौजूद है, जैसे your mitochondria, your lysosomes, vacuoles. Okay. Next, endoplasmic reticulum, Golgi complex. Like this, different types of cell organelles are present in the eukaryotic cell. Cell organelles are present. This is another difference. Cell organelles present. Okay, so there is only one cell organelle which you can find in the prokaryotic cell. So what is that? What is that? 70S ribosomes. 70S ribosome. 70S ribosomes are only the cell organelles you can find in the prokaryotic cell. Other than the 70S ribosomes, no other cell organelle is present. Understood? So next, another difference, the type of ribosome which you can see in the prokaryotic cells are the ribosomes are 70S type. 70S type of ribosomes you can see in the prokaryotic cell. Whereas in eukaryotic cell दो तरह का ribosomes पाया जाता है. एक है ATS ribosome and दूसरा है 70S ribosome. So two types of ribosomes you can see in eukaryotic cell. So these are the differences between prokaryotic cell and eukaryotic cell. Students, copy down this. Very important for your exam. Copy down. Students, now let us discuss about different shapes of cells. Cells differ in size, shape and activities. The cells differ in size, shape and activities. Just like we human beings, we occur in different sizes, different shapes and we do lot of different strange activities, right? Just like we human beings, the cells also, they show different sizes, different shapes and activities. Okay, so the shape of the cell vary with the function they perform. The shape of the cell depends upon the function which they perform. So now let us see different shapes of the cells. The first cell is RBC, red blood cell, also called as erythrocytes, which we can see in the blood. Okay, so if you observe the drop of blood under the microscope, you can see lots of red blood cells. The red blood cells, they have round biconcave shape. They have round biconcave like this. It is having concavity on both sides. So this is called biconcave shape. So what is the function of RBC, students? RBC helps in transport of oxygen and carbon dioxide, right? So transport of gases, oxygen and carbon dioxide, and they contain a red color pigment, it called hemoglobin. Very important. RBC contains a pigment called hemoglobin. So if you see the size of human RBC, the human red blood cell has a diameter of 7 micrometers, which is very important for your exam. Note down this point. Okay, next another cell, white blood cells. So in the blood there are another type of cells called white blood cells. They act as microscopic policemen. They protect our body from germs. Okay, the white blood cells, they have irregular shape. It is called amoeboid shape. They have irregular shape. It is called amoeboid shape. Then what is their function? Defense. They protect our body from disease causing germs. Next, the third type of cells, columnar epithelial cells. This we can see in all the animals. Okay, columnar epithelial cells, they are long and narrow. So most of the time these cells, they will cover the surface of organs inside our body. Columnar epithelial cells, and they are also found in the inner lining of intestine. You can see each and every cell is showing a nucleus, right? Now come to the fourth type of cell, the nerve cell, also called neuron. The nerve cells are present in our brain and also in our spinal cord and also in our nerves. Okay, so what is the function of nerve cell? Neuron, look here. The neuron conducts the nerve impulses. It will conduct the nerve impulses, the signals. Okay, the nerve cell is having branched long shape. It is having several branches which are radiating in different directions, and it is long, and there is a nucleus even in the neuron. They are found in the brain. Okay, next is trachea. What is that? Trachea. It is present in the plants. It is one of the plant cells. Okay, it is a dead cell, doesn't have any life. You don't find cytoplasm or nucleus inside this cell. The trachea has elongated shape with tapering ends, and there are several small openings on the surface which are called as pits. The function of trachea is to conduct water and minerals from root system to shoot system. So even though tracheids are dead cells present in the plant in xylem tissue, they can carry out a function that is transport of water and minerals. And what is their shape? Elongated. Next, the sixth type of cell, mesophyll cells. What are those cells? Mesophyll cells. Mesophyll cells, they are present in the leaf. They are plant cells and they can be seen in the leaf. Their function is to carry out photosynthesis. So formation of starch, food material in the presence of sunlight. Mesophyll cells present in the leaves, they have the function of photosynthesis. These cells are round and oval in shape like this. These cells have green colored cell organelles which are called chloroplasts. Chloroplasts. So chloroplasts are the kitchens of the cells. They take part in photosynthesis. That is about mesophyll cells. Next, mycoplasma. Mycoplasma are the smallest living cells in the entire living world. They are also a kind of bacteria, smallest cells and smallest bacteria. They doesn't have any particular shape. Such type of organisms are called pleomorphic organisms. They constantly keep on changing their shapes. They occur in different shapes. Okay, so that's why they are considered as pleomorphic. There is no definite shape in these organisms and they have the smallest size, that is 0.3 micrometers. Very important for your NEET exam. Mycoplasma, they are pathogenic bacteria. They cause a lot of diseases in animals as well as plants. Next, bacteria. Most of the bacteria, they have the size in the range of 3 to 5 micrometers. Next, coming to the largest cell in the living world, the largest living cell is the ostrich egg cell. Okay, so it is the largest isolated cell in the living world, that is ostrich egg cell. And which is the longest cell? The longest cell is the nerve cell. Nerve cell is the longest cell in the entire living world. So this is about different shapes of cells. So the shape of cells depends upon what, students? It depends upon the function they perform. RBC, since their function is to transport gases, they have this particular round and concave shape. And WBC, their function is to fight the disease causing germs. That's why this particular shape suits that particular function. So this is about different shapes of cells. Now let us see another topic. Students, now let us see the structure of prokaryotic cell. So what is a prokaryotic cell? It is a cell which is having a primitive kind of nucleus without any nuclear membrane. So there is a naked genetic material which is not covered by any nuclear membrane. We call that cell as prokaryotic cell. The prokaryotic cells are represented by bacteria, blue-green algae and mycoplasma. Okay. Blue-green algae are also called as cyanobacteria. They are photosynthetic bacteria. Mycoplasma are the smallest bacteria. Okay. Next, these prokaryotic cells, otherwise called as bacteria, they can multiply at very fast rate, even faster than eukaryotic cells. Literally within matter of 25 minutes, a bacterial cell can divide and produce 2 daughter bacterial cells. So that much fast they are in multiplication. Okay, they multiply rapidly than eukaryotic cells. Note down the second point. And coming to the shapes, there are 4 important shapes, 4 major shapes in bacteria. One is the rod shape. The rod shaped bacteria are called as bacillus, and spherical shaped bacteria are called coccus. Comma shaped bacteria are called vibrio, and spiral shaped bacteria are called spirillum, which we have already covered in the kingdom Monera. Next, coming to the structure of a prokaryotic cell. So here we are going to discuss a typical prokaryotic cell, which is a rod shaped bacteria. Okay, the prokaryotic cell has a rigid cell wall. A rigid cell wall. A cell wall gives mechanical support to this prokaryotic cell. And also it prevents bursting and collapsing of cell. It prevents bursting and collapsing of cell. So it is a rigid layer. It is made up of a material called as peptidoglycan. What is the material present in prokaryotic cell wall? Peptidoglycan. is the material present in the cell wall. Remember this. Okay. Now, below the cell wall, there is a thin membrane. What is that thin membrane called? Plasma membrane. Already we discussed plasma membrane. This plasma membrane, it interacts with the outer environment and help the bacteria to absorb the nutrients from outside. Okay, so this is the membrane which is able to interact with the outer environment. It is a semi-permeable membrane. What is semi-permeable membrane, students? It can partially allow certain molecules to enter into the body. It will not allow all the molecules to enter. So it is permeable only to some molecules. This nature is called semi-permeable nature of plasma membrane. So the structure of plasma membrane is same in both prokaryotic cells and eukaryotic cells. There is no much difference. It is almost same structure. Okay. Next, below the plasma membrane there is liquid. There is a fluid matrix. This fluid matrix is called as cytoplasm. What it is called? Cytoplasm. So this is the arena of different cellular activities. Several cellular activities will be taking place inside this liquid called cytoplasm. So in the cytoplasm lies our genetic material, the genomic DNA. The genomic DNA is circular. It is compactly packed. It is compactly packed. It is compactly packed in the cytoplasm, and this genetic material is not covered by any type of membrane. It is a naked genetic material. That's why we are calling it as nucleoid. So what is the region in which you can find this genetic material? It is called as nucleoid. Nucleoid is present. There is no nucleus. Okay. So this nucleoid represents the chromosome of DNA, which is circular. It is a circular DNA. Okay. In addition to this circular DNA, there are small circular DNAs. Small circular DNAs are also present in many copies. These small circular DNAs, they are called plasmids. They are called as plasmids. These plasmids, they control several phenotypic characters. The most important phenotypic character they control is resistance to antibiotics. Note down this point, students. Plasmids, they control a phenotype that is called resistance to antibiotics. What is resistance to antibiotics? They provide the ability to prokaryotic cell to survive against the antibiotics. Okay, so that is called resistance to antibiotics. So that is one of the phenotype conferred by plasmids. So these plasmids are also used in genetic engineering. So we will be discussing how they will be used in genetic engineering in coming chapter. Okay. So what else you can find in the cytoplasm? My dear student, what else we can find in the cytoplasm? In the cytoplasm we can find the non-membrane bound cell organelles, which are called 70S ribosomes. What are these? These are 70S ribosomes. और ये ribosomes क्या होता है बच्चों? Ribosomes are called protein factories. Ribosomes are called protein factories. They are involved in protein synthesis. Many 70S ribosomes, they are attached to messenger RNA. They are attached to RNA called messenger RNA, and they form a single unit. It is called polysome. What it is called? Polysome. So note down this, students. Several 70S ribosomes, when they are attached to messenger RNA, then this unit we call it as polysome, whose function is protein synthesis. It is involved in protein synthesis. Polysome can be also called as polyribosome. Polyribosome. What is its function? Protein synthesis. Okay. Now, plasma membrane, above the plasma membrane cell wall. Above the cell wall there is one more layer which you can see here. I have put here a dotted line. This third layer. What is this third layer called? It is called glycocalyx. Glycocalyx. Glycocalyx is another protective layer which is present over cell wall. Okay, so now look here, students. The plasma membrane, the cell wall, and the glycocalyx, all these three layers put together form cell envelope. They form cell envelope. So which will form cell envelope? Plasma membrane, cell wall, and glycocalyx, all of them together combine to form a cell envelope whose function is protecting the cell. Okay, even the individual layers have their own functions, but collectively they act as a protective layer which is called as cell envelope. Note down this point, students. What is a cell envelope? The glycocalyx is the outermost layer in the prokaryotic cell. In some bacteria, the glycocalyx can exist in the form of tough capsule. In some bacteria, the glycocalyx may be in the form of loose slime layer. Loosely arranged slime layer. Okay, the glycocalyx can be in the form of tough capsule in some bacteria. In some bacteria, it exists as loose slime layer. Okay, this is another point. So the beta, अब हम डिस्कस करेंगे बैक्टीरिया कैसे मूव करेगा. So there is a locomotor structure called as flagellum. It helps in movement in bacteria. The flagellum is a long whip-like structure. So because of the movement of the flagellum, the bacteria can propel itself in a particular direction. So in some bacteria you can find only one flagellum. In some bacteria you can find several flagella. Okay. So in some bacteria flagella may be absent. So there are different types of bacteria depending upon the arrangement of flagella. Okay. So what are the different parts in the flagella? The flagellum has a basal body. It has a basal body, a hook, and a long filament, a long part called as filament. So what are the three parts in a flagellum? The flagellum is having a basal body which is embedded in the plasma membrane, a hook which is embedded in the cell wall and glycocalyx, and a longest part is the filament. Which is the longest part in the flagellum? It is the filament. Okay, so this is about flagellum, locomotory structure in bacteria, prokaryotic cell. Next, coming to an important structure called mesosome. Mesosome. What is mesosome, students? You can see here the plasma membrane, it forms an infolding. This infolding formed by the plasma membrane is called as mesosome. The mesosome, it carries out several vital functions in the prokaryotic cell. Look here. So the mesosome shows the following functions. The mesosome, it takes part in cell wall formation. It also is involved in DNA replication, means doubling of DNA, and also it helps in distribution of DNA to daughter cells. The mesosome also, it helps in respiration process. It also helps in secretion process. It also increases the surface area of plasma membrane. And also increases the enzyme content present inside the cell. So these are all the different functions of your mesosome. Okay, so what is mesosome? It is an infolding of plasma membrane. Okay, the mesosome can exist in different forms. The mesosome can exist in the form of vesicles. Vesicles means round membrane bound structures, vesicles. They can also exist in the form of lamella, like this. These are the lamella. They can also exist in the form of tubules, tube-like structures. So mesosomes can exist in the form of vesicles, lamella, and tubules. So this is about mesosome, students. So in the prokaryotic cell, you can also find some reserved materials, some reserved stored materials will be present in the cytoplasm. They are called inclusion bodies. The reserve materials in the prokaryotic cells are called as inclusion bodies. The inclusion bodies will be like phosphate granules. In some bacteria you can find cyanophycin granules, glycogen granules. These are the reserve materials which you can find in the prokaryotic cell. So in certain photosynthetic bacteria called blue, green, purple and green photosynthetic bacteria. In blue, green bacteria and purple and green photosynthetic bacteria, we can see gas vacuoles. Gas vacuoles are non-membrane bound vacuoles. These gas vacuoles, they are not covered by any membrane, but they have some gas inside it. So because of this gas, what happens? The bacteria literally they are able to float in the water surface. So that is about gas vacuoles which are present in which bacteria? Blue, green, purple and green photosynthetic bacteria. Friends, let me erase a portion of the board. So look here students, in the bacteria there will be bristle-like structures which are on the surface, outer surface of the prokaryotic cell. These bristle-like structures are called as fimbriae. They are called as fimbriae. So what is the function of this fimbriae? They help the bacteria to attach to the solid surface. They help the bacteria to attach firmly to the solid surface such as rocks, and they also help in attaching the prokaryotic cell bacteria to the host cells. Okay, so these are the structures which help in attachment, fimbriae. So during the time of reproduction, they produce a tube-like structure, long tube-like structure which is called as pili. So by using this tube-like structure called as pili, the bacteria can transfer the DNA from one cell to the another cell. The genetic material DNA can be transferred from the existing cell to the another cell by using this tube-like structure called as pili. Okay, so in photosynthetic bacteria, a very important structure which is resembling the mesosome. Just like a mesosome, the infolding of plasma membrane, there is a structure called chromatophore. What it is called? Chromatophore. This chromatophore has lots of photosynthetic pigments. Photosynthetic pigments are present. So with the help of these pigments, the bacteria is able to synthesize the food material. So the chromatophores with pigments, photosynthetic pigments, take part in photosynthesis process in which bacteria? Photosynthetic bacteria. Okay, so that's all. This is all about prokaryotic cell, students. Copy down.